Hello everyone, today I am honored to introduce to you a study that comprehensive spatial profiling reveals transitional microbiome dynamics and microbial heterogeneity in pediatric adenoid hypertrophy. The cohort includes 101 adenoid hypertrophy, 119 adenotonsillar hypertrophy, 11 tonsil hypertrophy and 45 controls. Sample types are nasopharyngeal swabs, adenoid swabs, tonsil swabs, adenoid tissues and tonsil tissues, 1149 in total. All were sequenced by 16 srRNA. For 30 paired swab tissue samples, we performed metagenomics, qPCR, fish and bacterial culture. Principal coordinate analysis revealed that the bacterial community structures of the case group and the healthy control group were significantly separated, and different sampling sites also showed a distinct clustering trend. The Permanova test showed that the R-square was approximately 0.15, and the p-value was 0.001. This indicates that the anatomical location is a key factor in shaping the bacterial community of the upper respiratory tract. Each niche has its own dominant genus. Nasopharynx is dominated by Staphylococcus, mucosal surfaces by Streptococcus. Adenoid tissue shows a mixed profile with unique Ralstonia enrichment. The ternary plot confirms Staphylococcus from nasopharyngeal swabs, Streptococcus from surfaces, and Ralstonia enriched in adenoid tissue. In both adenoid hypertrophy and adenotonsillar hypertrophy, adenoid tissue microbiota clusters between nasopharynx and adenoid surface on PCOA, showing a transitional pattern. This suggests that adenoid tissue is influenced by adjacent niches and is not a simple pathogen reservoir. Metagenomic sequencing revealed that the four most abundant bacteria on the surface and in the tissues were Staphylococcus aureus, Haemophilus influenza, Streptococcus pneumoniae, and Moraxella catarrhalis. Strain-level analysis showed that the Staphylococcus aureus and Streptococcus pneumoniae from the surface and tissues of the same patient closer genetic relationship, suggesting that the bacteria could migrate within the individual. Fission culture also confirmed the presence of live bacteria in the tissues. We built models to distinguish mild, grade 1 to 3, from severe, grade 4, adenoid hypertrophy. The adenoid tissue model achieved AUC 0.752, while nasopharyngeal swabs and adenoid swab models had only 0.626 and 0.632. Tissue biota better reflects disease severity. SHAP analysis reveals that high Roseburia associates with mild symptoms, while Flavobacterium, Mycelia, Cotonella, Listeria, and Enterobacter are more common in severe cases. These are potential microbial markers. Summary: firstly, we have established the largest-scale spatial microbial map of children's adenoid hypertrophy, covering multiple ecological niches from the nasopharynx to the adenoid tissue, and from the surface to the deep part. Secondly, the sampling site significantly affects the microbial community structure. Adenoid tissue shows a transitional dynamic and has tissue-specific enrichment. Thirdly, multi-platform verification suggests that key bacterial species can persist both on the surface and in the tissue, possibly participating in the maintenance of chronic inflammation. Fourthly, the microbial community in the adenoid tissue has a higher predictive for the severity of the disease than the surface swabs. These findings provide an important foundation for future multiomics integration and targeted microecological intervention.